Hi guys, my name is Wyatt Jensen. I am a state 4-H ambassador and a Jackson County 4-H'er, and today I wanted to talk to you guys a little bit about a very important topic when it comes to agriculture, specifically whenever we're talking about livestock, and that is animal and livestock genetics. So today I just wanted to break down a few of the main terms you'll hear when talking about livestock genetics and how we might figure out a few key details as well. So first up, for the importance of livestock. Anytime you're judging show animals of any kind, you're always looking for soundness, finish, muscle, and structure. All of those things can be attributed back to genetics. So that is why it is so incredibly important, and obviously each of those things affect the breeding quality of livestock and the meat quality of livestock. We're going to get into a few of the main terms as well as how we figure some practice problems in, say, Punnett squares, for example. So as you can see, I've got 5 terms screen shared here, and we're just going to go through and kind of simplify and define all 5 of those terms. So the first one here is dam, D-A-M, and all that really means is just the mother. The sire. The next one is the father for livestock. The next one I have here is a gene. And all that a gene really is, whenever you think about it, is just whatever trait is being passed on and then being expressed. So it's just a characteristic or a trait. And then this next one is called a Punnett square. And it's used very commonly in genetics to see what the possible outcomes are. So all I'm going to write here is chart that shows genetic outcomes. And then another one that I wanted to talk about, we're not going to really dive too deep into this in this lesson, but it's called a pedigree. And all that really is a family tree for livestock. And we'll commonly see that when we're looking at herds or flocks over a long period of time. So now before we get into how Punnett squares work or how pedigrees work, I just want to talk about a little how we represent genes and how we write them down on a piece of paper. All right, so talking about how to notate different genes in your livestock. So here I've got an example, dam and a sire as always. And in the dam has black hair in this scenario, and the sire has red hair. So say this is cattle, and we've got an Angus dam and a Hereford sire. So to get started off, we want to try to communicate and represent those genes in a way that's efficient, but everyone knows what we're talking about. So starting off with the dam has black hair. To represent this, you have to choose a letter. It can be any letter of your choosing as long as you know that it represents black hair. So for this example, let's just say B. And so I wrote a capital B and a lowercase b. And so for the red hair, let's do the exact same thing. And let's just choose R for red. And I wrote a capital R and a lowercase r. Now, why did I write a capital and a lowercase of each of those letters? When we're talking about genes, we will always have a dominant gene and a recessive gene. And the way that we notate a dominant gene is with a capital letter, and the way we notate a recessive gene is with a lowercase letter. Now, what do those terms mean? All dominant means is whenever it is present, it will always express itself. So if you have a black-haired gene and say it's dominant, then it will always express itself. You will always have black hair if you have a black-haired gene. For red hair, say that it's recessive. If you have a recessive red-haired gene, it means that you could be red-haired. However, if you have a different dominant gene, then it will kind of stay back. It will recess, hence the name. So with that going forward, we're going to be using those letters, and I'll talk about it a bit more as we use those letters again. But we're going to use that to show how we cross genetic material whenever we're talking about livestock. Okay, so I've gone ahead and written out the first example. As you can see here, remember we've got a dam and a sire, a mother and a father. And when we combine those two genetic materials, we, in this case, have a dam that is an Angus with blue eyes and a sire that is a Hereford with brown eyes. And so for this specific example, we are going to be talking about eye color, and I'm going to show you guys how you can even predict what the possible outcomes are for eye color of your baby calf. First off, when we're talking about eye color, in the middle of this screen here, I've got a black box, and it's separated into 4 smaller boxes, and that is called a Punnett square. And so I'm going to show you guys how to fill it in. So how do we notate blue eyes? Well, if you remember back a second ago when I said that we always have either a capital or a lowercase letter for each one, and it either has to be recessive or dominant. So for this example, how do we distinguish which one is recessive or which one is dominant? Well, we really don't know unless we find out for ourselves. So now it's common knowledge that blue eyes are a recessive gene and brown eyes are a dominant gene. But scientists have to test this multiple times to figure out which one is truly the dominant recessive gene. Nowadays, we have this stuff figured out, and you can usually look it up to see if particular genes are dominant or recessive. So starting out here, I'm just going to tell you that for the dam, blue eyes are a recessive gene. So for the dam's blue eyes, we're going to have a little b and another little b. Because blue eyes are a recessive gene, I'm just going to tell you that starting off the bat. And so we have two lowercase b's, which means blue eyes. And for the sire has brown eyes. So how do we represent brown eyes? Well, there are two different options. We could either have a big B for brown and another big B for brown, or we could have one big B and a little b. Now, why is this? A second ago, remember when I told you that the recessive gene, if it has another dominant gene with it, will not express itself. So in this example that I have right here, B for brown, big B for brown, and a lowercase b for blue, when we have those two together, the reason why it's not a mix between blue and brown is because blue is recessive. So with the dominant gene, it recesses back and does not express itself. So now that sire could either be a purebred, brown-eyed sire, or it could be brown-eyed with a recessive, or it could be a carrier for blue eyes. And when you talk about more genetic diseases, you usually use more terms such as carrier and things like that. But for this example, let's just say that the sire has purebred brown eyes, big B, big B. And so, with that being said, we're going to use this Punnett square here to represent the different outcomes of crossing the dam and this particular sire together, what those outcomes could be. So for the dam's genes, she's got a little b. So I'm going to put one little b over one row here, and I'm going to put the other little b over another row here. All right, now for the sire's genes. We've got two big B's, so I'm going to write one big B here in this row, and another big B here in this row. And now we've shown what the two parents' genetic information is. And when we cross it, it looks a lot like a multiplication table, where you take the top and you multiply it by the bottom, or the row. And so the column and the row together make, and here we don't have to change any other letters, they make just a big B and a little b. And the same here. We've got a little b and a big B, so it just makes big B, little b. And here I'm color coding it, but normally you wouldn't always have to do that. And also notice that I'm always putting the dominant gene first, so we can definitely tell that it is present and it is expressing itself. And so again, on the next two boxes, we're going to have the same result, big B and a little b. Big brown B and a little blue B. All right, so that example, I know it might not seem easy, but we're going to do some more examples, and you guys are probably going to better be able to do this on your own. Okay, I'm going to move on, and I'm going to give you guys an example problem, a practice problem. And you guys are going to pause the video and try to solve it for yourself. If you don't get the right result, no worries, just try to solve it along with me afterwards. Okay, so here we go. I'm going to write it out for you guys. And we have a dam and a sire, as always. In this example, let's say that the dam has a spotted tongue. And let's say that the sire has a solid colored pink tongue. All right, so with this example, let's say that the spotted tongue has gene, let's say S. So it has gene S. And let's say that the spotted is recessive to the solid color. So I'm going to write this here in black. The spotted tongue will be 2 lowercase s's because it is recessive. And let's say that the solid pink color is dominant. So I'm going to write this here in pink, 2 capital S's. Normally we don't like to pick letters that kind of look the same whenever they're written lowercase and uppercase. But in this example, I'll try to make it as clear as possible. Just remember that anything in pink is a dominant gene, and anything written in black is a recessive gene. All right, so now I'm going to write out my Punnett square. Okay, so I've got my Punnett square, and I've written the genetic information out to the top and the side, and I want you guys to find what the results could be. Are they all the exact same? Are all outcomes the exact same? Or are there some different outcomes? And what are the chances that I will get a spotted tongue? And what are the chances that I will get a solid pink tongue? Right now, I want you guys to pause the video, and I want you guys to try to solve this problem on your own. Okay, so hopefully you guys have solved the problem, and what result you should get is a big pink S and a small black S in each box. You should have gotten the exact same thing in each box. So we've got a 100% chance of a dominant pink gene and a recessive spotted gene. And what does that mean? Well, remember, whenever a dominant is present, it always takes over. So the pink is present in every single scenario, so we know we have to have a 100% chance of getting a pink tongue. So it was a very complicated way of thinking about it. However, I want you guys to be able to apply this to your own herds or your own livestock if you have them. If not, don't worry about it. But you guys can also come up with some different examples at home. Thank you all for watching this video. I hope you guys learned something. If at any point it was too complicated, you can always rewind, or I know YouTube has several other resources on this as well. Try to look it up, try to do your own research. That's perfectly fine. And also, I hope you guys can check out some of the other videos here on this virtual clover page if you have not already. Again, thank you guys for watching, and see you guys next time.